Hi, my name is Amit Scheer. I'm a recently graduated undergraduate student from the University of Ottawa, and I'm going to be presenting our lab's work on identifying human biological processes and protein sequence motifs that are putatively targeted by SARS-CoV-2 proteins. Gordon et al. Recently published the SARS-CoV-2 interactome, a map of human host proteins interacting with SARS-CoV-2 viral proteins upon viral infection, by individually expressing 26 out of 29 SARS-CoV-2 proteins in HEC293 human host cells and performing affinity purification mass spectrometry, the authors identified 332 high-confidence protein-protein interactions, or PPIs. As protein-protein interactions are crucial in defining host-pathogen interactions, this interactome contains a wealth of data that can be analyzed to determine biological insights into SARS-CoV-2 infection. For example, knowledge of the gene ontology terms and protein sequence motifs that are overrepresented and locally enriched in these PPI networks can help to identify disease mechanisms and new therapeutic targets. There exist numerous approaches for the analysis of the clustering of proteins in PPI networks. One type of approach involves first breaking a network into smaller clusters using an algorithm such as Markov clustering, and then querying each cluster for functional and motif enrichments. Another type of approach involves annotating each node in a PPI network with its associated functions or motifs, and then analyzing the clustering of each set of annotated nodes as implemented in algorithms developed by our lab, including GoNet and LESMON. Here, we present a set of functional enrichment and protein sequence motif analyses to thoroughly investigate the set of human host proteins interacting with SARS-CoV-2 viral proteins upon infection. First, we performed functional enrichment analyses of the PPI networks of each viral protein by querying each PPI network for overrepresented Go terms using Ontologizer. We found that human proteins purified by SARS-CoV-2 viral proteins were enriched for certain Go terms. For example, NSP13 interacted with proteins that were involved in the microtubule organizing center and interciliary transport, whereas ORF8 associated with proteins involved in the extracellular matrix and the endoplasmic reticulum lumen. Human host proteins interacting with each SARS-CoV-2 viral protein can also interact with each other. To provide this context to the viral host PPI networks, we added human PPIs from the string database to produce what we refer to as the string-augmented network. We then obtained clusters of the string-augmented PPI network using Markov clustering and analyzed each of these clusters for the enrichment of Go terms using Ontologizer and protein sequence motifs using MEME. Using this approach, Markov clustering and Ontologizer identified Go terms that are significantly clustered in the augmented PPI network. These included, for biological processes, regulation of nucleotide metabolism, establishment of RNA localization, and cilium organization. Similarly, MEME identified protein sequence motifs that are enriched in Markov clusters. However, these motifs occurred primarily among homologous proteins, therefore this was not an unexpected result. As a different method to detect locally enriched Go terms and sequence motifs, we applied GoNet and LESMON-Pro, modifications of algorithms that had previously been developed in the lab, to the string-augmented PPI network. First, the nodes in the network are annotated using the Go terms for GoNet, or sequence motifs for LESMON-Pro, that are associated with the protein situated at that node in the network. Then, the shortest path distances are calculated for each pair of proteins in the network to produce a distance matrix. That distance matrix is applied in the calculation of a total pairwise distance for each annotation of the network, which is the sum of the shortest path distances between each pair of proteins annotated by that Go term or sequence motif. The clustering significance of that total pairwise distance is then assessed using a Monte Carlo sampling approach, which produces a p-value. Finally, a false discovery rate distribution is produced by repeating this analysis on a shuffled version of the network. Since this algorithm relies on the assessment of clustering in the entire network, rather than first breaking the network into smaller clusters for analysis, we expect that this algorithm will be more robust to sets of homologous proteins. Similarly to Markov clustering and Ontologizer, GoNet identified Go terms that are significantly clustered in the network. For biological processes, this included cell projection assembly, organelle localization, and nuclear transcribed mRNA catabolism, among other Go terms. Our algorithm to detect locally enriched protein sequence motifs, LESMON-Pro, identified numerous protein sequence motifs that are significantly clustered in the network. These motifs were then hierarchically clustered into 9 families, each represented by a motif containing degenerate characters. These degenerate motifs were then searched against the protein sequences of all proteins in the PPI network in order to generate the sequence logos displayed here. This figure shows proteins that contain sequence matches to each of 6 of the 9 representative protein sequence motifs and the location of these proteins in the PPI network. While LESMON-Pro still tended to uncover sequence motifs that annotated multiple proteins in the same family of homologous proteins, such as nuclear pore proteins or RAB proteins, the majority of the motifs also annotated several non-homologous proteins. The fact that these motifs were associated to several proteins in the network and that these proteins were significantly clustered within the network suggests potential functional relevance for these motifs. Our results provide insights into the function of SARS-CoV-2 viral proteins. For example, better understanding of the interactions between NSP13 and proteins related to microtubule organizing center could lead to new therapeutic targets related to NSP13. Similarly, ORF10 has not been highly characterized thus far due to its limited sequence homology with other proteins. Therefore, the fact that its interactors are enriched for ubiquitin ligase complex proteins could provide insights into its function. Locally enriched protein sequence motifs could also be involved in host-pathogen interactions. For example, study of a motif shared by 2 proteins that are known to directly interact and regulate innate antiviral immunity could provide insight into how SARS-CoV-2 disrupts these cellular processes. Another motif that's associated with highly clustered proteins involved in protein trafficking could provide clues as to how SARS-CoV-2 components traffic upon viral infection. Finally, we envision that further study of the identified protein clusters, Go terms, and sequence motifs could provide insights that could help to identify the disease mechanisms of SARS-CoV-2 infection. To conclude, I'd like to sincerely thank my supervisor, Dr. Mathieu Lavallee-Adam, as well as my colleagues in his lab, specifically Rachel, Sarush, Emily, Dallas, Irina, and Joshua, for their immense contributions to this project. Thank you very much for listening.